This figure, I'm going to talk a little bit about how many hormones, especially the hydrophilic ones, are stored within vesicles prior to their release. And the rationale for this storage is that it dramatically speeds up the response time of particular endocrine tissues when a stimulus is present. Right? Because in many cases, hormones are made from transcription translation, right, like peptide and protein hormones, or through enzyme modification of precursor structures, such as those that come from amino acids. In either case, that can take some significant amount of time and delay a response. So instead, in many cases, we actually produce the hormones in advance, store them in vesicles, and then have the stimulus simply result in the release of that preformed hormone. So how this works, for the peptide and protein hormones, which are already going to pass through the Golgi apparatus in the process of their production, we end up having them bud off of the Golgi within vesicles. And what we find is that these vesicles are then labeled with a protein called Vsnare, V for vesicle. And the snare proteins are adapted to aid membrane fusion. So basically what they do is they target packages of hormone to a particular membrane target. So in the case of an exocytosed hormone, right, that membrane target, of course, would be the cell membrane as opposed to an intracellular organelle. What we find is that there's a transport distance here. And so our vesicles are transported using what's called a motor protein, right? This little green blob here sort of grabs our vesicles and marches down a microtubule, the cytoskeleton, and then sort of like creating this pool of vesicles just underneath the membrane. And those end up being tethered to the actin filaments of the cytoskeleton, just sort of holding them in place essentially as a reserve. Now, when our stimulus arrives, what happens is the V snare and the T snare come together and aid the fusion of the membranes, the vesicle membrane and the cell membrane, right? And so our T for target, right, is on the inside of the cell membrane, ready to bind with our V snare protein. Now, when our stimulus arrives, for all regulated exocytosis, the stimulus will result in an increase in intracellular calcium levels. And that's simply the last trigger. That's the last thing we need, right? We're all ready to go. Our V and our T snares are already like lined up and ready to do their job. And the calcium is the trigger for the membranes to fuse and dump our vesicle contents, our hydrophilic hormones, into the interstitial fluid. Right? So our hormones end up out here. They then move by diffusion into the bloodstream where they're carried off to their target tissues. Now, one of the byproducts of all this vesicular exocytosis is that the cell membrane would expand sort of endlessly if we didn't do something about it. And so what happens is we actually endocytose the membrane, add it back to an endosome and back to the Golgi, essentially reversing exactly what happened with our exocytosis of the vesicle over here. Now, one thing you might note over here on the right is that our steroid hormones do not undergo this process. They are lipophilic hormones, and they are readily diffusible through the cell membrane and can enter the bloodstream directly. This also means, because this is an enzymatically produced hormone, right? It's a hormone produced from the cholesterol precursor, that the response time to steroid hormones may be a bit slower. Finally, there are some hormones that are hydrophilic that are not peptide or protein based, not coming from the Golgi, such as the catecholamines. In that case, the vesicles will actually take up the precursor molecules. The hormones are formed within the vesicles, and then they follow the same pattern as the peptide and protein hormones being exocytosed.